Hey, Hammy here, coming to you with the first part one of section 3.3 on cell transport. Specifically in this video, we're going to look at passive transport. Okay, what is passive transport? Passive transport, like if something's passive or if a person is very passive, you might think they're not very energetic. Okay, so the first part here, passive, two keywords, okay, energy, without energy. Okay, so passive means substances are going to be transported or moved across the plasma membrane without the use of energy. Okay, most of the time they're going to go from where there's a lot of them in a certain area, so we call that concentration, how much of something per volume. So where there's a lot of things or high concentration to an area of where there's not as much or a low concentration. Okay, one of the simplest types of passive transport is diffusion. Okay, we've used that term before and maybe not thought specifically about what exactly it means. Okay, things diffuse, they move across the membrane or from one area to another because of differences in concentrations. Think about grandma baking bread. Okay, and you walk into her house, or fresh cookies or something like that, and you walk into her house, you smell that freshly baked bread, okay? Because those bread molecules, okay, molecules are naturally in motion, okay? And around the oven, right where that bread is baking, there's a lot of those bread molecules. And they'll slowly ping pong off the other molecules in the air, and they slowly spread out, right? Until it fills the whole house. And when you open the doors, it'll start to spill into the outside. Okay, with that, the smell from the bread. Okay, same thing happens in our cells. So here's our plasma membrane or our lipid bilayer, and you see that there is a lot of stuff out here. So this would be our high concentration out here. Notice in here, nothing, right? So this would be our low concentration. So high concentration, low. These things will just naturally kind of be moving, shimming, shaking around, and they'll slowly go between the phospholipids, okay, until you get kind of an equal concentration on both sides. They'll go till it equals out. Think of a clear beaker of water and take a drops of food coloring in it. What happens? Okay, it slowly spreads out, and if you'd let it there all day, okay, without doing anything to it, just the natural motion or the natural energy and motion of the molecules, it would slowly spread out evenly. Your beaker would be all nice, the water would all be the same color. Okay, so now in simple diffusion in and out of the cell, it has to be things that can go through the lipid bilayer, okay, or the plasma membrane. Okay, so you remember the hydrophilic heads right here are polar. They're hydrophilic, they're water loving because there's water out here and there's water in here. Okay. And then in the center here, it's non-polar tails, the carbon to carbon to carbon to carbon to carbon bonds of the fatty acid tails of that phospholipid. So it's non-polar here in the middle part. So the only thing that can really go between the phospholipids are hydrophobic or non-polar molecules. Okay, this happens, a very important process in humans is respiration, right? Breathing. Okay, when you breathe in oxygen, oxygen will come out of the air into your lung cells and eventually into red blood cells, into the hemoglobin where it's taken out to your cells. Carbon dioxide is brought back, okay, leaves your tissue cells and comes back, okay, because there's a high concentration. Okay, so O2 or oxygen and carbon dioxide, CO2, are examples of little molecules, tend to be very small, nonpolar, that can go between the phospholipids. The second type of passive transport is osmosis. Osmosis is a special type of diffusion. It's the diffusion of water across a semipermeable membrane. Most of the time, that being our plasma membrane. Okay, so essentially it's water moving in and out of the cell based on the concentration. Now, there are three terms that I want you to be familiar with. You have both of these pictures in your notes, and I want to talk a little bit about them. We're going to add some stuff to it. Over here, these are animal cells, okay? And this picture over here are plant cells, okay? In animal cells, if the concentration is the same on the inside and outside of the cell, okay, that is called isotonic. Now, notice down here at the diagram, does that mean there's no water going in and out of the cell? No, there's still water moving, but notice the yellow arrow going in and the green arrow coming back out. It's the same amount of water. So isotonic means the same concentration inside and outside of the cell. There's no net movement of water. Water is moving in and out, but the cell is not swelling or shrinking or anything. Okay. In the first example over here, hypertonic. Hyper, I remember hyper because what makes you hyper? Lots of sugar, right? So lots of sugar, think lots of sugar, lots of solute. Now, why doesn't, if the, okay, in the environment out here, so this would be in a hypertonic solution, okay? So let's say we put it in in salt water, put a red blood cell in very salty water, okay? Can the salt go into the cell? No. So if the salt or the sugar can't go into the cell, water will move out of the cell to try to dilute that concentration. So what happens to the red blood cells? Okay, they shrivel up, okay? That that cell shriveling of an animal cell, and I want you to write this down, is called crenation, okay? Crenation. And they just shrivel up. And if you could take blood cells, put them in salt water under a microscope, you'd see them all shriveling up like this, okay? In an isotonic solution, remember the concentration is the same, so the blood cells will stay the same. There's equal movement of water in and out. Notice hypertonic solution, the water is moving out of the cell. Hypotonic, okay, now there's more salts and stuff inside the cell. It can't leave the cell, so notice there's net movement of water into the cell. What's going to happen? Okay, they'll swell up with water, and an animal cell doesn't have a cell wall, so they can actually burst. We call that cell bursting cytolysis. Cyto meaning cell, lysis meaning burst. Okay, so it's cytolysis. Okay, the cell will actually burst. If you, let's say that this was distilled water. If you put red blood cells in distilled water, that distilled means it doesn't have any solute in or very, very little. And now the cell is going to be more concentrated. Okay. And the water will move in and you could actually see them burst. Okay. A plant cell is a little bit different because what do you notice? Kind of the dark blue here in navy color. Yeah, they have a cell wall. Okay. So if you place a plant cell in a hypertonic solution, again, not the cell, we're talking about the solution. The water will leave the cell. Notice the cell wall stays there, and the plasma membrane shrinks away from the cell wall. Okay, this is called plasmolysis, or we say the cell's plasmolized. The term is called plasmolysis. Okay, plasmolysis. So the cell walls stay there, and the cell just kind of sucks in like this. The cell membrane, sucks in and leaves the cell wall behind. Remember, isotonic means equal. Okay, so there's equal movement. The cells stay the same. Hypotonic. Okay, notice that there's a movement of water in, but they're not going to burst like animal cells because it's just going to apply pressure to the cell wall. And see how the cell wall is kind of pushed out? Okay, we say that the cell is turgid, or we call it turgor pressure. Okay, and turgor pressure is what keeps the cells up. And I talked about this in the previous video. And if you don't water your plants for a while, they'll start to wilt. And so when you give them water, they'll fill back up again, right? So that is called turgor pressure. So please know that hypertonic, isotonic, and hypotonic, and know the difference in what happens in different solutions with animal versus plant cells. Okay, and we'll review this in class as well. The last type of passive transport is facilitated diffusion. Okay, facilitated or facilitator means a helper. So this is diffusion across the membrane with the help of a transport protein. Okay, so these are proteins that are embedded in the phospholipid bilayer that will help molecules go through. Now remember we said if we look down here at this membrane, this middle layer, we said is hydrophobic. So non-polar molecules like O2, oxygen, and CO2 can just go in between there. Other small molecules that are charged or hydrophilic, and some biomolecules like glucose and amino acid that our cell needs to get in or out, okay, cannot go in between through that hydrophobic region. Okay, so facilitated means they need a little help. They need an opening to get through. Okay, that can be one of two things. It can be a channel protein. Okay, the channel protein you see right here, it's just embedded right within the plasma cell or plasma membrane, has a hole through it. So if you look at it, look like a donut. Okay, and you can see that the molecules will go, okay, can go in and out. They're still going to go down the concentration gradient. Okay, they're still going from high concentration to low. Okay, so high concentration of little, let's say this is glucose because they're kind of like hexagon shape. Okay, if there's a lot out here, then the glucoses will start moving into the cell. Okay, other things like say this is an amino acid. Sometimes they have what's called a carrier protein. All the carrier protein does is there's three steps. So number one, it's open. Okay. Let's say that this is an amino acid right here. The amino acid will kind of fall into that little spot that it fits in there. So step two is it's right here in the middle. And then step three, it expels it to the interior. This is intracellular space. Okay, so it's going into the cell here. Okay, and these things can often work in reverse order. If there's a high concentration inside the cell, they'll go out of the cell as well. Okay, so facilitated transport, you're still going down a concentration gradient. Okay, but they have little helper molecules for things that might be charged or hydrophilic. Some of the biomolecules that we talked about in chapter 2. Okay, so remember, these are all passive transport. Moving down the concentration gradient, it does not need any energy from the cell. The molecules just naturally spread out from high to low concentrations. In the next video, we'll take a look at active transport. What happens if we already have a high concentration and we want even more? Okay, the cell then needs to expend energy to do that, and we'll look at that in the next video.